Hello everyone, my name is Muhammad Danish Mirza bin Muhammad Ali Reza, a student from University Technology Malaysia, and today I will be presenting on my research titled Effects of Il bulbosa derived compost and commercial fertilizers on Il bulbosa growth and soil microbe composition. With the increasing demand of sustainable agriculture and circular farming, my study investigates the potential of turning Elytrian bulbosa plant waste, specifically its leaf and bulb parts, into compost-based fertilizers. Using 16S rRNA amplicon sequencing, we evaluated the effects of this compost alongside BSF compost, NPK, and a control on both rhizosphere microbial communities and plant growth. We found that Il bulbosa-based compost enriched beneficial microbes like Caulifigura corniformis and supported better plant traits. Functional analysis also showed improved nutrient cycling under organic treatments, highlighting the potential of this compost to support sustainable and circular cultivation. Modern agriculture heavily relies on synthetic fertilizers. While they boost productivity, they also contribute to soil degradation, microbial imbalance, and water pollution. As an alternative, sustainable farming emphasizes recycling waste. Il bulbosa, also known as bawang dayak, is a medicinal plant rich in bioactive compound, but generates unused bulk and leaf waste. So this research explores whether that waste can be turned into compost to serve as a circular, eco-friendly alternative to chemical fertilizers. There are three main objectives in this study. The first one is to compare the rhizosphere microbial communities under different fertilizer treatments using 16S rRNA amplicon sequencing. The next objective is to analyze the predicted functional roles of those microbes in relation to plant growth. And the final objective is to evaluate the impact of each treatment on the growth and yield of Il bulbosa. So this study involved 5 treatments, which is NPK fertilizers, commercial BSF compost, Il bulbosa leaf compost, bulk compost, and also a control. So the plants were grown for 6 weeks with fertilizers applied once in 2 weeks. At harvest, rhizosphere soil was collected and sent for 16S rRNA sequencing to analyze for microbial profiles. We also measured growth parameters like plant height, number of leaves and tillers, SPAD value, as well as yield parameters such as bulb number and weight. Data were analyzed using microbiome analysis, STEM for pathway prediction, and also statistical tools like ANOVA and Duncan test. For the results of my research, starting with alpha diversity represents the diversity within a single sample or environment and is a key metric for evaluating microbial ecology. 2 diversity indices were focused on, which are the CHO1 index, which estimates species richness, focusing on the number of different taxa, including rare ones, that are present in a sample, and also Shannon index, which accounts for both richness and evenness, reflecting how evenly distributed individuals are among those taxa. BSF compost treatment showed the most balanced and also diverse microbial community based on its Shannon index, while NPK had the highest richness CHO1 index but with lower evenness, indicating microbial dominance by certain species. For beta diversity, which reflects the differences in microbial community composition between samples or treatment groups, the Bray-Curtis PCOA plot showed that NPK samples clustered distinctly from the organic treatments. This means NPK significantly altered microbial community structure and also compared to organic amendments. Looking at the relative abundance of the microbes, at the phylum level, Planktomycetota was highly abundant in bulk compost, while firmicutes, especially Lactococcus, dominated in NPK-treated soils. And at the species level, Caulifigura corniformis was more abundant in bulk compost, whereas Lactococcus dominated in NPK, indicating a microbial imbalance. For functional prediction, STEM analysis showed that organic treatments, especially bulk compost, enhance beneficial pathways like ECA cycle, sulfate assimilation, and also nitrate reduction, which are important for nutrient cycling. In contrast, NPK promoted pathways related to fast microbial growth like purine and peptidoglycan biosynthesis. This indicates functional divergence aligned with the taxonomic shift discussed earlier. In terms of plant growth performance, BSF compost produced the highest overall growth rate and bark compost produced the most bark, interestingly, while leaf compost resulted in the tallest plants. This suggests that each organic treatment supports different growth rates, possibly due to the microbial function they enrich. So, in conclusion, E. bulbosa-derived compost show great promise as effective organic fertilizers. They supported beneficial microbial communities, enhanced plant performance, and align well with the principle of circular farming. This research provides evidence that plant-based waste can be recycled into functional inputs that support both sustainability and productivity, especially in medicinal plant systems. Before I end this, I would like to express deepest gratitude to my supervisor, Dr. Huzalina binti Hussein, and also my co-supervisor, Dr. Raihana binti Rizwan, for their continuous guidance and support. I also would like to thank UTM and UGS for this opportunity, and to IURC2025 for allowing me to present this work. That's all from me. Thank you for your time.